You can see my screen?
Yes, it's all good.
Okay, great. So it's
Thanks.
nice to follow on this interesting talk by Sylvain, because here is another system, co-evolutionary system, that builds diversity by this very piecewise fashion, uh, which I think is the way biology can generate a lot of diversity. So let me first acknowledge, oops, sorry, why is this happening? Let me first acknowledge my collaborators, mostly responsible for the theoretical work. My previous postdoc, Shixin He, now a faculty at Purdue University, and my previous postdoc, Shai Pilosov at Ben-Gurion University. So my talk today is about negative frequency dependent selection in Plasmodium falciparum malaria, arising from competition for host as the result of specific immune memory. So here patterns of immune protection and cross-protection generate competition as a function of antigenic similarity, and frequency dependence confers an advantage, of course, to rare antigenic variants and a disadvantage to common ones, promoting coexistence and diversity. Now, interestingly, for ecologists, frequency-dependent interactions are an important hypothesis for coexistence in another hyperdiverse system, namely the tropical rainforest. And I'm interested here in parasites that evade immunity by assembling their antigenic repertoires from multi-gene families and multi-gene families with a vast pool of variation. So I see these parasites as the rainforest of epidemiology. And although I will focus on Plasmodium falciparum and Plasmodium falciparum in high transmission regions, similar strategies are found in many other pathogens, other protozoa, but also in bacteria and fungi. And in Plasmodium falciparum, the main antigen of the blood stage of infection, this protein PFEMP1, once expressed, is exported to the surface of the red blood cells. And each parasite has 50 to 60 genes encoding for this protein within this family known as the VAR genes. So from the perspective of here our models, a parasite, a strain will be a repertoire, a combination of these VAR genes. These genes are expressed sequentially, and if there is memory from previous exposure to the product of any of these genes, then expression does not occur. Expression is shortened for that type. So what changes the fitness of the parasite here is the duration of infection as a function of the overlap with your memory. And I mentioned the very vast diversity of these multi-gene families in high transmission regions. This is an example from data by my colleague Karen Day and collaborators. These cumulative diversity curves and the contrast, for example, here on the left, this is from sequencing the markers of these genes. And you can see that in low transmission regions here in the green and blue, you can reach perhaps 100 different types, while in West Africa, you would find in these high transmission regions that these cumulative curves are going up. And in our recent studies in Ghana, in northern Ghana, you can see these curves accumulating to 10,000 and sometimes more. So you can do the combinatorics if you were choosing combinations of 50 from this kind of variation. Um, Now, the mechanisms, a major mechanism of diversity generation for different VAR genes is ectopic recombination during the blood stage of infection, with two different genes can recombine to produce true novelty. There is also meiotic recombination, which shuffles repertoires and generates then diversity at the level of repertoires, and this happens inside the vector. So what I would like to do today is to, in this topic of from models to data, to talk about our, first about our earlier work on this question, can we tell whether negative frequency dependent selection is indeed structuring parasite populations? And I mean, the question is, given this large diversity and these high rates of recombination, how can we tell whether there is structure in empirical data? In essence, what macroscopic patterns can we use to identify an important role of such process? And then in the second part of the talk, I will get back to theory, because if this is happening in this large, as I said, space of variation, then we want to ask what sustains, and how can we modify, how can we interfere with this high diversity? Because after all, it's important to notice that this variation, this antigenic variation, is important to oppose essentially successive intervention. What we, in these high transmission regions, we know that that elimination and control are extremely difficult, and with different results than in low transmission regions. So I have this, I like this slide. Some of you have seen it before, just to point out that the structure of the repertoires is not random in local populations. This is a study, again, with my colleague Karen Day. And you can see here, from samples they obtained from the blood samples of every child in a village in Gabon, where the isolates of the repertoires of VAR genes, essentially the isolate of VAR genes of each child, are lined up in different rows and columns. This quantity, the pairwise type sharing, is just measuring the degree of overlap. And all you have to see is that, of course, the overlap is very low, meaning that each child has its own set of infections. And of course, you can say, well, given the high diversity, this could occur at random. But if you do randomizations here on the right, and you measure the mean, the expected, well, you look at the mean overlap compared to the distribution from this randomization, there is a force here that is creating some limiting similarity. But of course, talking about non-random structure is not the same as asking about non-neutral structure, because in the end, this is, we have the demography of transmission that to consider. So in our work, we built a computational stochastic agent-based model. I will not describe it in detail here, but basically the important point is that we can keep track of the memory and the history of exposure of each individual person in the population, and we can have the different processes that you expect in this vector transmitted system, the ectopic recombination, etc. So again, I will not describe the model, but what I wanted to point out in this from models to data is that we can use these computational models to essentially to compare the output and the patterns, compare the full model that has this immune memory, specific immune memory, to the patterns we expect under neutrality. And we have two types of neutrality: the complete neutrality, in which this aspect of fitness, duration of infection, is not affected by, is just a constant as a function of age, and a second type of neutrality in which essentially we have this general competition for host, because only the number but not the identity of previous infections influences immunity. So the patterns we used were not trees or phylogenies because of recombination. So we relied on network representations of repertoire similarities. Essentially, the networks, the nodes are repertoires, and the links basically are measuring in a bidirectional way the similarity between the repertoires. And of course, this would be a completely connected network, but because we are interested in the structure of competition, we kept the tail of the links, essentially the structure of the network that for the parasites that would be competing the most strongly because of their similarity. And I will not go into details here. What we have found is that we can basically look at an ensemble of properties of these networks of the order of 30 different ones that characterize different aspects of the networks, from motifs to degree to different components, etc. So we use this ensemble of properties to build a classifier. So we use our simulations within parameter ranges for these high transmission regions to say, can we classify essentially the simulations to the right process? So we build this classifier, and indeed we can differentiate. The important point here is that in theory, negative frequency dependent selection in other parasite modeling before was shown to show coexistence of non-overlapping repertoires, basically clusters. But at this very high diversity of the genes themselves, we can no longer look for those simple patterns. Nevertheless, there is a signature of limiting similarity in the networks. And we can also look at networks and their similarity over time with multi-layer networks. And the point I wanted to make here was that there is a property that differentiates from the neutral case. In both cases, there are modules which can be considered strains, but the modules persist much longer in the presence of frequency-dependent selection, which retrospectively is not surprising because balancing selection should promote persistence. Now, from models to data, the important point to us was now we have macroscopic patterns in the networks that give away a structure of limiting similarity. So we apply it to sequence data from our study site in collaboration with Karen Day and her team. And without going into details, I wanted to point out that this classifier uses this method, this multivariate method of analysis of genetic data, known as discriminant analysis of principal components. The details do not matter. It is a really nice method for structure of genetic data. And here you have in different colors, the different dots are the different simulations assigned from the to the different neutral regimes in blue and yellow, and the red are the simulations generated by selection, by frequency-dependent selection. So you can really separate those regimes, and the dot here is the data for 2 seasons where we can classify it to immune selection. So again, this allows us to ask this question. There is no simple clusters arising from competition for hosts, but there is a degree of limiting similarity that is evident in the networks. Now let me now transition quickly to this second part, just to say how is it that we live in this space where we then have such high diversity, genetic diversity, from which to assemble the repertoires. So we look here, what we propose here is the existence of a threshold for the accumulation of innovation. And this applies to the malaria system, but would apply also to other parasites, and in particular parasites with multiple antigenic sites, multiple epitopes, etc. But the basic idea is illustrated in this graph. Of course, if new genes are generated at a rate that is too low compared to their persistence time, then we don't accumulate them, And on the right, we have the case where that rate is sufficiently fast compared to their time. So we should have a number that we called R diversity, this reproductive number that multiplies a rate of generation to the rate of the typical lifetime of the genes. And if this is bigger than 1, then we will accumulate innovation. In the paper, Shishin derived an expression for G nu, which depends, of course, on n, the rate of generation mu, but also the probability of invading the system, and then T nu, which can also, for which we don't have a complete analytical expression, but only an approximation, because the solution of this comes from a nonlinear PDE. But we have for G nu an analytical formula, and then we can get T nu from just from the simulation. So what I want to show you here is essentially an illustration of this threshold, a verification of this threshold in the simulator. What you have is the logarithm of this number we propose, RDIV. So the threshold is at 0. Percentage of new genes during an arbitrary time. You set a time, and then you say how many of the genes are new. And you see these dots are different simulations with different assumptions of within host dynamics, different parameters, seasonality, no seasonality, and you see that indeed such threshold exists. You can say, we know that the pool of variation will decrease with transmission intensity, but how does it decrease? Interestingly, there is a monotonic increasing relation between RDIV and the intensity of transmission, which in malaria is measured by EIR, the entomological inoculation rate, which is the number of infected bites per unit of time. But what is important here then is that if we vary the intensity of transmission, we can push the system below this threshold. And I should say, for those of you who are wondering, this is well above R0 of 1. So these systems are all transmitting. The question is, can they accumulate diversity? And this is an illustration of this behavior, where basically you see on the left below threshold, on the right above threshold, the different colors are the frequency of different genes that are coming, new genes coming into the system. You see the accumulation on the right, and on the left, the gray are just the aggregate of low frequency genes, but you see some genes coming and going in what looks apparently neutral. Essentially, the variation cannot stick. Again, for R0 above 1. Now, I will end by saying that these some beginning results of how this matters, we are convinced that this kind of, how can I say, invisible threshold that we have ignored does matter for perturbations and intervention. These are simulations. The colors in the bars show that these 4 simulations differ in whether we have, by decreasing transmission, whether we have crossed this threshold or not. Only the green has crossed transmission. And you see here on top that if you, at time 0, we basically decrease transmission. If you are above threshold, after intervention, you continue accumulating new genes. While if you have crossed the threshold, you don't. And if you look at the central panel at the bottom, you see that the behavior of the prevalence is different, whether you have crossed or not. In particular, if you have crossed RDIV, you basically cannot rebound. And I should say that very often in the interventions in high transmission regions, we observe rebounds and that really oppose intervention. As the system is not just recovering prevalence, it's recovering diversity, which allows it to escape immunity. So we believe that this threshold is really important in terms of more successful intervention once you have crossed it. So I'm going to speculate for ecological systems in general, just to end, by saying that hyperdiverse systems may well occur at the opposite end of neutrality. with coexistence assembled under specific and frequency-dependent interactions. This would set the stage for the existence of an unappreciated threshold that concerns the accumulation of the genetic variation on which high biodiversity is built. And we would not know we have crossed this threshold and transitioned to increased fragility of the system. So the question now back to data would be, how do we determine in a practical way in epidemiological systems, whether such as malaria and these very diverse systems, that we have crossed this threshold, because we would be putting them in very different conditions. Let me end by, well, I will not have time for this, by acknowledging my collaborators on the empirical side. I mentioned Karen Day and here other colleagues in her lab. Thank you very much.
Thank you very much, Mercedes, for the great talk. So now we have